Thank you. So the brief today is talk on NTRK fusion testing, and there are multiple receptors. 3 of them, the NTRK receptors, and they're encoded by specific genes, NTRKs 1, 2, and 3. They're expressed in human nervous system, and they play an essential role in the physiology of development and function of the nervous system through activation by neurotrophin. So that's going to be the ligand which binds to the receptor. The TRK receptor signaling leads to cell proliferation, differentiation, and survival. The applied aspect of this thing is that if they have abnormal NTRK fusions, they may act as oncogenic drivers. The TRK gene family can drive the cancer progression after recombining with other genes such as ETV6, EML4, and so on and so forth. These proteins are aberrantly expressed and constitutionally active, so they don't need the trophin to actually stimulate the receptor. The receptor is always switched on. Something that needs to be kind of emphasized here is when there's a fusion between the NTRK genes, the kinase domain is always contributed by the NTRK, and the extracellular ligand binding domain is the partner gene. So you can see that here. This is the NTRK3 here, and that's the PTK, that's the tyrosine kinase domain being contributed by the NTRK, and the fusion partner ETV6 contributes the extracellular ligand binding domain. Now, for oncogenic activation of these genes is typically caused by in-frame fusions, and I'll come back to what's that in-frame fusion, leading to expression of a chimeric protein, which always includes the kinase domain, as I just mentioned in that. Why is in-frame fusion important? For NTRK at least, if the fusion maintains a reading frame of the downstream gene, then the amino acids will be in frame, they will be in the correct sequence, and the protein which will be expressed will be active. If you have an out-of-frame gene fusion, there's a frame shift. The amino acid sequences go on changing, and it's not a productive protein which is being developed. Now, NTRK rearrangements need to be potentially in frame to have a confirmed fusion partner to actually have that oncogenic potential. Now, not all NTRK rearrangements lead to a functional gene fusion product. These non-functional fusions are defined as rearrangements. So if you're going to act on it, you need to have an NTRK fusion and not an NTRK rearrangement. Now, different NGS testing platforms will either tell you that it is a fusion or a rearrangement. At least the Foundation in one report says that. But otherwise, you actually need to do immunohistochemistry to find out if the protein is actually being expressed or not. Now, there are various technologies available for fusion detection. That's IHC, there's FISH, there's real-time PCR, and there's NGS. Everything has some advantages and disadvantages. As far as immunohistochemistry is concerned, it's low cost, it's easily available. Most labs would have it. It's a rapid turnaround time. And the antibody that one has, it's a pan-NTRK antibody. It tests for both, all three rather, NTRKs A, B, and C. Now, the disadvantage is that it may not be specific for NTRK gene fusion, because the antibody will pick up a wild-type protein as well as a fusion protein. It may possibly cause false positive because tissues which are rich in neuronal tissue, such as, for example, smooth muscle tumors or neurogenic tumors, may anyway express NTRK. So you don't know that. As we are confirming for lung tumors, it may not be applicable, but in general, IHC for that reason has a low specificity. Well, there's no standardization of scoring algorithms as yet. As far as FISH goes, again, availability is easy. The location of the target is kind of visible. You look under the microscope into that particular cell, but it requires knowledge of any of the fusion partners because you're using a fusion product. But the problem is that you'll have to have 3 separate FISH testing for the NTRKs A, B, and C. So that's the problem. Chromosomal translocations can sometimes result in false positive signals, and you might also get good amount of false negatives. That's part and parcel of the FISH analysis. Real-time PCR is good. It's high sensitivity and specificity. It's kind of low cost. The issue is that we must know the target sequence because you're putting up labeled primer sets or probes towards those genes. So you can't possibly identify the novel fusions. As far as NGS goes, it will detect all possible fusion partners. It can be used to evaluate multiple actionable targets simultaneously, so it's useful. It gives you information beyond NTRK also. RNA NGS is preferred over the DNA because in RNA you don't have the introns, so you have more chances of pickup. Now, the problem with DNA NGS is that it's limited by intron size, but the RNA itself has a problem. Sometimes the tissue is not well fixed and the RNA quality is poor, so you may not get any of these answers. Now, pan-TRK IHC testing varies with the kind of fusions that one obtains, so you can get a nuclear positivity, cytoplasmic, or a membrane. So that's useful to actually determine what kind of a fusion exists. And these are images of a fish, which I borrowed from somebody. This is not something which my lab does, but you can get, you can use a fusion signal, and you can get a fusion signal and two separate individual, the arrows which are pointing towards that NTRK fish. Now, designing the NGS panel is important. Now, if you have a wider panel, if you use single gene panels versus small limited panels, the chances of pickup are less. But if you have slightly more wider panels, it basically incrementally increases the chances of a NTRK fusion detection. You need to have a multi-parametric approach to identify NTRK fusions. So at least in terms of lung tumors, because the possibility of obtaining is less than 1%, what should drive you to actually analyze it? You typically get it with adenocarcinomas with no driver mutations that drives you towards hunting for an NTRK fusion. If you do immunohistochemistry is usually cytoplasmic, because usually the NTRK3 ones are the ones which give you a nuclear positivity. You should persevere in terms of a driver negative lung adenocarcinoma. In terms of immunohistochemistry, it should be a diffuse and very bright expression for NTRK. And if you're using an NGS panel, prefer an RNA NGS, because that's more likely to yield you an answer. Now, NTRK fusion is commonly seen in adenocarcinomas, but sporadic reports also have been mentioned for squamous carcinomas, neuroendocrine carcinoma, and sarcomatoid carcinomas as well. The common NTRK gene rearrangements are the NTRK1 and NTRK3 rearrangements. The numbers are decimal. It's ranging from 0.07% to 3.3, which is slightly on the higher side for NTRK1. Otherwise, 0.02 to 0.2 and 0.08. So it's less than 1%. So your chances are a needle in a haystack. Now, NTRK fusions usually tend to present in a mutually exclusive manner with other canonical fusions and mutations. So therefore, one should identify, if you're looking for NTRK, it's a driver fusion negative lung adenocarcinoma. Now, although these are reported mostly in middle-aged patients and with a non-smoking history, and that's usually seen with other fusion-driven cancers, there have been reports which you've seen them in smoking individuals as well as elderly patients. Now, interestingly, NTRK fusions are also identified as a resistance mechanism to EGFR TKIs, particularly the fusion with the gene TPM3. So to summarize, to me, basically, in real world, you need to use either IHC or NGS. You can use IHC to screen it. If it's positive, it needs to be confirmed by NGS. Sometimes even by NGS, if the report doesn't tell you if it's in frame or otherwise, you might need to use the IHC again to check if the fusion protein is expressed. Now, different diagnostic tests and algorithms can be used. You need to identify the advantages and disadvantages. And I'll just run through these pros and cons of IHC and NGS diagnostic testing again. So NGS, high specificity, high throughput. You can multiplex and use multiple targets with that. You require a low sample requirement because it's a small input tissue which gives you more information. The cons are that it's expensive and a longer turnaround time vis-a-vis immunohistochemistry, which can be rapid. You can do it in a couple of days. Although there's no chance of multiplexing with IHC because it's a specific pan-NTRK IHC. It's inexpensive, easily available. The issue is about the low specificity, and it may identify samples which are not really NTRK because it will identify the wild-type protein, and the specificity is limited. So that's it from my side. Thank you very much for a patient hearing.
Thank you, Dr. Jay.